Hello again. I hope you are doing well wherever you are. Welcome to today's session. Today we'll be covering receptors of special sensation. Remember our last video that we did was receptors of general sensation, and our presenter is as shown there. So let's go to the scope for the session today. We are supposed to define what is a sensory receptor. We're supposed to understand the classification of various receptors, but for objective number 2, I will kindly refer us to our last video that we did on general sensation, where we discussed the receptors, their classification in terms of functional classification, and also structural classification. And then for today's session, we're going to describe the receptors of special sensation. So what is a receptor? A receptor is a cell or any structure that can be able to perceive sensory sensation and then can also be able to transduce. So remember, there are about 5 to 6 characteristics of a sensory receptor. Once again, I will refer you to the video on general sensation for us to know what are these characteristics of a sensory receptor. Then for what is a sensation? A sensation is the ability for a cell or this structure they're calling a receptor to be able to perceive a stimuli. So the awareness of a sensory stimulus is now these sensations. And they can either be two. You can have spatial sensations or somatic sensations. Somatic sensations are general sensations. Then spatial sensations are now the ones that have 5 characteristics that you are going to define and see in this slide. So there are 5 in number, and none of them is that they are localized receptors within the head and neck region. So these their receptors are localized in the head region. So that is cephalization. Then the other distinct characteristic is that they have spatial sense organs. They have distinct spatial sense organs. For example, the eyes, that is where you'll find the consonant rods. The tongue, that is where you'll find the taste buds. The nose, that's where you'll find olfactory epithelia. Ears is where you'll find now the hair cells. And then all those are innervated by cranial nerves. For example, the eyes are innervated by the optic nerves. Tongue, innervated by cranial nerve number 9 to cranial nerve number 11. That is glossopharyngeal, vagus, and cranial accessory. Then nose, that is olfactory nerve. And then ears is a vestibulocochlear nerve. And then just to wrap it up is that there are 5 special senses. And these 5 special senses include olfaction, gustation, hearing, vision, and equilibrium. So olfaction for the nose, gustation is now for the tongue, for taste, hearing, and equilibrium is for the ears, and then vision is for the eyes. Yes. So we are going to classify these special sensations in terms of they have a functional. They are actually when you classify special sensations, you do it functionally. And how do you do it functionally? You classify them according to the stimuli that they perceive. So for example, chemoreceptors. So chemoreceptor, that means there are some chemicals that are being perceived. And these are senses of taste and smell. So when you taste the food, when you smell the good odor, the bad odor, is due to those chemical substances which have been sensed by the chemoreceptors. So the taste buds and the olfactory epithelium, they are chemoreceptors. Then mechanoreceptors are now these receptors that can be able to perceive change in equilibrium and then hearing. So that is mechanoreceptors. And then to finalize up is you have photoreceptors. So photoreceptors which can be able to detect changes in electrical energy in terms of the lights, electromagnetic waves. That is the sense of vision is due to that ability of the cones and rods, which are photoreceptors in nature. So we'll start us off by looking at olfaction. And for olfaction, one of the best that you need to describe is this olfactory epithelium. And olfactory epithelium is actually as you see there. So here is actually the top of your nose, the roof of the nose, and also the sides, the upper 1/3 of the nasal cavity, and also the nasal septum will contain the olfactory epithelium, as you can see here. It has various cells, which we'll describe in a few minutes. Then it will have olfactory nerves, as you can see there. Then you have the cribriform plates, and then we can appreciate, here you have the olfactory bulb, and then you can appreciate the mitral cells, which act as cells of synapse. And as you can see, is that after you have the olfactory bulb, you have the olfactory tract. So that is the olfactory epithelium. And this olfactory epithelium is actually pseudostratified columnar epithelium without goblet cells, and it's ciliated. So this is pseudostratified columnar epithelium without goblet cells, but it's ciliated. The difference between it and respiratory epithelium is that respiratory epithelium has goblet cells, which produces mucus. And it has three major cells. It has olfactory neurons, or not the olfactory cells, which are now the cells that can be able to conduct. As you can see here, these are the olfactory neurons. Then you have the supporting cells and the basal cells. So supporting cells just offer support in terms of nutrition, um, Then the basal cells, which are now give rise to other cell types. For example, give rise to now these olfactory cells, and also give rise to the supporting cells. That is the basal cell. So any receptor has basal cells. Any special sense organ has basal cells or supporting cells. And then it has now these special receptor cells, which are now conduct the sensation. So for example, now we have olfactory neurons, supporting cells, and basal cells. So those are the three cell types in the olfactory epithelium. As you can see in the next slide, is that this is the olfactory epithelium, and this olfactory epithelium is pseudostratified columnar epithelium, ciliated without goblet cells. As we can see, there is no goblet cells. You can see this is a cilia. These are pseudostratified columnar epithelium, and then you can be able to appreciate. You can be able to appreciate. You can be able to appreciate that these are the neurons, and then the supporting cells are in between, as you can see there. And then you have the basal cells. These are the nuclei for the basal cells. Now you'll go ahead and talk about the olfactory nerves are present here. Then these are the Bowman's glands. These are the Bowman's glands, as you can see there, which are serous in nature. And these are the olfactory nerves. So that is the olfactory epithelium. What are some of the disorders for olfaction? Some of the disorders for olfaction is that you can have anosmia. Anosmia is absence of the smell, like complete absence of the smell. And then hyposmia is that there is diminished sensitivity of the smell. So anosmia is complete absence of this sensitivity of the smell. Hyposmia is now diminished sensitivity of the smell. Dysosmia is now distortion of the normal smell. Yes, the smell is there, but it's distorted. And then you can have others such as phantosmia, where you have now the perception of a certain odor, which is completely absent. So perception of odor in absence of another stimulus. So for example, the smell of a pizza, you are perceiving the smell of a pizza, and yet the pizza is not present. So that is phantosmia. And then parosmia is now distortion of odor perception, and another is present. So the odor is present, but now the perception is absent, or the perception is distorted. That is parosmia. So the five were the most common ones that you'll be able to appreciate is anosmia, hyposmia, dysosmia, then phantosmia and parosmia. You can be able to go them at your own time, maybe for general knowledge. The other one that we'll go ahead and talk about is actually gustation. And gustation now is the tongue. So the tongue is a special organ, And the tongue is the one that give us the taste of the smell, as you can appreciate here. So we have this is a tongue, as you can appreciate, this is a tongue. Here you have the epiglottis, as you can appreciate there. Then we have this is a palatine tonsil. Then the palatine tonsil, you can appreciate it has these are palatine bed, palatoglossus and palatoglossus and palatopharyngeus. These are palatine bed. These are, you can see the palatine tonsil. Then we have the lingual tonsil. Remember the lingual tonsil and the palatine tonsils, they make up the Waldeus ring. So we can discuss the anatomy of the tongue in a few seconds. Is that the tongue is divided into two parts. We have the anterior 2/3, and then you have posterior 1/3, and then you have now the dorsal, the root of the tongue. So the anterior part of the tongue has the anterior part of the tongue and the posterior part of the tongue are divided or are separated by this boundary, the sulcus terminal, which is V-shaped. And anterior to it, you have some lingual papillae, which we'll discuss them in a few minutes. Then the posterior 1/3 is actually divided, has the lingual tonsil, and this lingual tonsil makes up the Waldeus ring, as you'll see in a few minutes. So the tongue now is a sense organ for taste, and then these taste receptors that are found in the taste buds, of which now the taste buds are distributed in the tongue, the pharynx, and the larynx, and even some parts of the oral cavity. And then they're clustered into taste buds. And then we have now the dorsal surface of the tongue being divided into two by the sulcus terminalis. So these are anterior 2/3 and the posterior 1/3 being divided by this sulcus terminalis. So the anterior 2/3 is the oral part, which has lingual papillae, and then the posterior 1/3 is the pharyngeal part, which has now the lingual tonsil. So the posterior 1/3, which has lingual tonsil, has immune functions. Then the anterior 2/3 is the oral part, has now the lingual papillae, and then these lingual papillae are the ones that will contain the various taste buds, as we'll see in a few minutes. So we'll go ahead and describe the 4 types of lingual papillae. As you can see them, here is our tongue. And the tongue, the anterior 2/3 of the tongue, has various lingual papillae. And there are 4. One of them is this circumvallate papillae, then you have foliate, you have fungiform, and then you have filiform papillae. So these lingual papillae are the ones that will contain now the taste buds that will contain the receptors specifically for gustation. And we'll start with, we have filiform papillae. So these are filiform papillae. Then you have the fungiform papillae, as you can see it here. We have the circumvallate papillae, as you can see it here. And then the last one is the foliate papillae. So these 4 will contain taste buds, but one of them will not contain taste buds, as you will see in a few minutes. So filiform papillae, this one thing to appreciate is that it doesn't contain taste buds. And then in addition to that, it provides friction that facilitates movement of the food during chewing. Then for the other lingual papillae, which is fungiform papillae, this one will contain taste buds. So as you can see them here, these are the taste buds. It actually looks like mushroom. Fungiform papillae contain taste buds. And a point to appreciate is that this fungiform papillae will contain about 5 taste buds each. So maybe it can be 5 plus or minus 1 for each fungiform papillae. Then another point to appreciate is that this circumvallate papillae, which is not just anterior to this sulcus terminalis, is the largest papillae. There are about 10 of them in number, and then they will contain about 100 taste buds each. So that is circumvallate papillae. This is circumvallate papillae. Actually, this circumvallate papillae, and then it will contain taste buds. Foliate, the fungiform papillae will contain taste buds, and then the foliate papillae, which is now rudimentary in human and may be absent in aged individual. So it's actually rudimentary in human and may be absent in aged individuals. This is the foliate papillae. Now, all those will contain taste buds, and this is the taste bud. So in our next slide, we're going to see the cell types. Or we are first of all going to appreciate the histological presentation of this lingual papillae. As you can appreciate here, this is a filiform papillae, which doesn't have taste buds. Then you can appreciate fungiform papillae with the taste buds being present. Circumvallate papillae with the taste buds on the walls. And then we have now the foliate. This is the foliate papillae with the taste buds, as you can appreciate. So these foliate, filiform, fungiform, and then circumvallate. So this is what you can be able to appreciate when we look at this lingual papillae histologically. So a point to appreciate, how does this lingual papillae come across? For more information regarding this, kindly visit our video on development of the tongue and how it comes across, how you develop these lingual papillae. And now we have the tongue. Yes, we have the tongue, the anterior to that of the tongue. It has various lingual papillae. We have seen these lingual papillae, they are four in number. For example, we take the circumvallate papillae. So this circumvallate papillae, it will have taste buds. And then these taste buds will contain cell types. So what are these cell types that are found in these circumvallate papillae? As you can see here, there are three cell types. And one of them is this gustatory receptor cell. And then, as I told us earlier, is that we'll have a supporting cell, and then also we'll have a basal cell. So there are three cells in number, as you can appreciate here. So this is a gustatory receptor cell. And then we'll have now the supporting cell, and then we finalize with the basal cell. So these are test buds, as you can appreciate in this slide. So these test buds, first of all, will appear like they are onion-like, they are oval. So you can see they have onion-like in terms of they have many layers. They are oval, as you can see here. They are pale staining, unlike the surrounding region, and then they extend the full thickness of this tongue, the masticatory epithelium, and then it opens to the surface through the taste pole. So this is a taste pole, as you can appreciate here. This is a taste pole, this is a taste pole, and that is now the taste buds and the cell types, as you can appreciate there. So what are the various categories of various taste buds? There are about 5 of them. We have sweet for sugars and related compounds, salty for sodium compounds, and then you have now sour for compounds or various foods that will contain hydrogen ions, for example, from acids. Then bitter taste for these foods that will contain alkaloids, and then additional human taste sensations, for example, umami and water sensations. So umami are these foods that will contain amino acids, small peptides, and nucleotides. And then water is now ideally water sensations from water being detected by water receptors in the pharynx. So sweet sensation, salty sensation, sour, bitter, umami, and water are some of the taste sensations, but 4 of them are primary. Then you have additional ones. The primary ones are sweet, salty, sour, and bitter. Then the additional ones are umami and water sensations. So these are the various categories of taste buds, and at which point of the tongue are they perceived. So for example, you can appreciate the umami. For example, this is now the tomato. It's perceived by mostly the anterior part of the tongue. Then the tip of the tongue is actually the sweet part. And then you have now the lower part of the tongue, of the anterior part of the tongue is actually salty. This sensation, then sour is actually now on the lateral aspect of the tongue. Then bitter is actually now the posterior part of the anterior part of the tongue. That is now bitter sensations. But now that doesn't rule out that various lingual papillae with taste buds will be located in those regions. But that fact is, this lingual papillae will be located everywhere in the tongue, in the anterior two-thirds of the tongue. And then you can perceive any taste buds, any category of these taste buds anywhere. But now this is a classification that was used in the olden days. So we go ahead and describe the various cell types in the taste buds. As we've seen, we've talked about supporting cell type 1. Then you'll have now gustatory cells, which are now from type 2 to type 3. And then you have the basal cells. So supporting cells, as you can see here, these are the supporting cells. This is a supporting cell. The supporting cell. And this supporting cell is actually an immature cell. It's slender and elongated, as you can see here. Immature cells, slender and elongated. And then it is involved in clearing of neurotransmitters. And then also things to do with nutrition and removal of waste through these neurons. Then for the gustatory cells or the taste receptor cells, which are from type 2 cell type to cell type number 3. Remember, type 1 is supporting cell, type 2 and type 3 are now gustatory. Receptor cells are also elongated. You can appreciate them here, they are elongated. They have apical ends with microvilli. And then this microvilli projects towards the taste pole, as you can see it here. And then these microvilli now are the ones that interact with these receptor taste stimuli receptors. And then the last bit to appreciate is that type 2 receptor cells taste sweet, bitter, and umami, which is G-coupled receptors. And then type 3, which taste sour and salty, are ion-gated channel receptors. So type 1 cell type is supporting cell. Type 2 and type 3 are gustatory cell types. All these cell types are elongated, but gustatory cell types, they have microvilli projecting towards the taste pole. The reason for this for interaction with the taste stimuli. Type 2, which ideally at this point tastes sweet, bitter, and umami, is a G-coupled receptor. And then type 3, which is sour and salty, perceives sour and salty sensations, ideally is ion-gated channel receptors. Then the last cell is now the basal cells, which are now the stem cells for the other cell types now, type 1, type 2, and type 3 cell types. To finalize is that you're supposed to appreciate the histology of these taste buds. How do they look like? As you can appreciate here. Here is a taste bud. Here's a taste bud. This is a masticatory epithelium. The masticatory epithelium. Most likely this is circumvallate papilla. You can appreciate this is a taste for and the various cell types. These various cell types you can appreciate you have. Now the basal cells here, the supporting cells, and then you have the neurons at the same. So as you can appreciate here, the taste for cell type 1, cell type 2, and then now type 3 cell types, and then type 4 cells, and now the basal cells, and then here we have the nerves. So the disorders for taste is that you have ageusia. Ageusia is complete absence of taste. Hypogeusia is the diminished sensitivity of taste. Dysgeusia is the distortion of the normal taste. So, and then you have now others, of which we won't cover them today, but a point of appreciation is that the 3, ageusia, complete absence of taste sensation. Hypo means it's less, diminished sensitivity of taste. Then dysgeusia, this means like something that is distorted, distortion of the normal taste, dysgeusia. Then they are far more prevalent in elderly population. So they are prevalent in more of the people who are aged. And then this typically, the olfactory perception is more likely disrupted during the aging process than the sense of taste. So as you can appreciate here, so this is a pathway for gustation, as you can appreciate here. So cell nerve, the nerve, cranial nerve number 9, 10, and 11, as you can appreciate there. So, and even the facial nerve, you can appreciate them there. Now they go ahead and they carry the decussate at the brainstem. Then they go to the thalamus, and then they go to the various olfactory areas, as you can appreciate there. So, but the point of appreciation is that the disorders for taste is ageusia, hypogeusia, and dysgeusia. So kindly make sure you go through them at your own time very well. We go now to the third special sensation, which is ears, the vestibular auditory system. So this vestibular auditory system is that the ear has dual sensory function, maintenance of equilibrium, and also maintenance of, and also the perception of sound, that is hearing. So receptor for hearing is the organ of Corti. The receptor for equilibrium is macula and crista capillaris. And this organ of Corti and macula and crista capillaris, they are found in this labyrinth. And this labyrinth are two. You can have bony labyrinth and membranous labyrinth. So as the bony covers the membranous labyrinth, as you can see here. So this is ear, this is outer ear, this is middle ear, then this is inner ear or the internal ear. So the internal ear has bony labyrinth. So this is a bony labyrinth, the one in brown. Then the one in blue, that is the membranous labyrinth. So the bony labyrinth, the central space is known as the vestibule. So this is a vestibule. And then now it will have this vestibule will give rise to three semicircular canals, which are now three semicircular canals posteriorly, and then anteriorly it will give you the cochlea. So laterally, the vestibule is separated from the middle ear by two fenestrations. So these two fenestrations, the oval window and the round window, as you can see there. So the oval window is occluded by the base of the stapes, which is one of the ossicles, and then the round window is occluded by membrane similar to the tympanic membrane that is present between the external ear and the middle ear. So that is the bony labyrinth. For the membranous labyrinth, as you can appreciate here, is that this vestibule contains the utricle and the saccule. And then now this semicircular canal shaft will be found posteriorly, and here you'll have the cochlea. So these are the membranous component, membranous labyrinth. They're housed by body labyrinth, as we mentioned earlier. They'll contain endolymph. So these semicircular canals, utricle, saccule, cochlea will contain endolymph. And then this membranous labyrinth is separated from body labyrinth by this perilymph. Perilymph separates the body labyrinth from membranous labyrinth. And then it will contain five structures, as we see. So you'll have the utricle here. Then you'll have the saccule. The utricle and the saccule will be contained in the vestibule. Then the lymphatic ducts opens into the saccule. And then you'll have the cochlear ducts. Here's a cochlear duct, which opens in the saccule. And then the semicircular duct, which open in the utricle. So for more information on how we have development of these structures, kindly visit our video on development of the ear, embryology, on how we develop the utricle, saccule, semicircular canals, and even the cochlea embryologically. So this is it. You can appreciate here we have a cochlea, we have the saccule, this is a utricle. Then you can appreciate at this point 7 semicircular canals. So at the end of these semicircular canals that have some enlargements known as crista ampullaris. And then this crista ampullaris will contain some specific cells that we'll see in a few minutes. Then in addition to that, you can appreciate this cochlea. The cochlear ducts has, if you can cut it transversely, you can appreciate it has three chambers, and these three chambers has various fluids, and also it has a receptor for equilibrium, as we'll see in a few minutes. We can start with this at this point, is that we have on the crista ampullaris, we have this crista ampullaris. You can see it's a magnified one. And this crista ampullaris has various cell types. And these cell types are now type 1 and type 2 hair cells. And then not forget that you'll have supporting cells, and also it will also have the basal cells. So that is now the crista ampullaris. So macula and crista ampullaris will contain type 1, type 2 hair cells, supporting cells, and basal cells. For the cochlea, it will have this, and actually this one you can be asked in an exam setting. You can be told to draw a cross-section of the cochlear duct and name various chambers and the various structure that are found in it. So for example, suppose to appreciate this is now the bony labyrinth, and then this is the membranous labyrinth. The membranous labyrinth is divided into scala vestibuli, the cochlear duct, and the scala tympani. Uh, the Heuser's membrane separates the scala vestibuli and the cochlear. And then this scala tympani is being separated from the cochlear duct by this basilar membrane. But you see an important structure here, which is now the spiral organ of Corti. And this spiral organ of Corti will contain the various cell types, and then it will also contain the cochlear nerve, which supplies it there, as we'll see in a few minutes. So cell types, two basic cell types. We have sensory hair cells, and now we have, so these sensory hair cells are the receptors specifically. Then you have the support cells, and these supported cells will have about 7 cell types. So the hair cells are classified into type 1 and type 2. By this point, we call them inner and outer hair cells, which are columnar in shape. They can be able to transduce sound energy into electrical energy, so that is hearing. And then they have stereocilia on the apical ends. And then their bases don't reach the basilar membrane. The rest of pharyngeal cells, these pharyngeal cells are one of the supporting cells. And then their base is in contact with peripheral neuron, now the cochlear nerve, being from the fibers of the cochlear nerve. For the support cells that will have now the inner and outer pharyngeal cells, also known as cells of Daytas, inner and outer pillar cells, also known as rod cells, cells of Hensen, cells of Claudia. So if you can count them, there are about 6 supporting cells. So inner and outer pharyngeal cells, inner and outer pillar cells, cells of Hensen, cells of Claudius, and now you add the hair cells. So an assignment to us is that we can go ahead and go and check what are the differences between inner and outer hair cells in terms of their modification, and then also in terms of their number, the frequency, the modality, and everything regarding this inner and outer hair cells. Some more important for us. As you can appreciate here, we had alluded earlier that this is scala vestibuli, contains perilymph, scala tympani contains perilymph, and then this cochlear duct. This cochlear duct contains now the spiral organ of Corti. It will contain endolymph. And then this cochlear duct, a point to remind us is that this cochlear duct is a scala media, and then it contains endolymph. A high magnification of this organ of Corti, can say it on our right side, is that you can be able to appreciate there's a membrane above this organ of Corti, spiral organ of Corti, which is known as tectorial membrane. Can be able to appreciate that this is one inner hair cell, then 3 outer hair cells. We have a basilar membrane, fiber of the cochlear nerve, then you have the various supporting cells. So these are the various pharyngeal cells now at this point. These are the various pharyngeal cells. Then you have pillar cells and other cells. I'll see. So go ahead and check what are the various cell types and what are their modifications towards supporting these hair cells. Histologically, how will it look like? We can appreciate here scala vestibuli, scala media, scala tympani. You can appreciate this is the vestibular membrane or the Hauser's membrane. And be able to appreciate this is a basilar membrane. Then this is now the tectorial membrane. You can appreciate this is spiral limbus. And then you can be able to appreciate this is spiral ganglion actually. This is a cochlear nerve. And then be able to describe that this contains perilymph, endolymph, and then this also contains now the endolymph at this point. Perilymph, endolymph. And then this is the bony labyrinth. Then the remaining structures that are found inside are the membranous labyrinth. Yes, so a higher magnification of the same, you can be able to appreciate the tectorial membrane. You can be able to appreciate the olfactory nerve. You can be able to appreciate you have 1 inner hair cell, you have 3 outer hair cells. These are the supporting cells, as you can appreciate them here. Yeah, so that is a spiral organ of Corti. This is scala media or the cochlear duct, scala tympani, and then this is scala vestibuli. Vestibular membrane is here, and then this is a basilar membrane, as you can appreciate that. So then, what are some of the disorders with hearing? Is that the disorders of hearing can either be sensory neural or conductive deafness. Sorry, neural deafness or conductive deafness. And for sensory neural deafness is that it's caused due to damage to the sensory receptors in the inner hair, in the inner ear. So we talk about the hair cells in the organ of Corti or the auditory nerve leading to the brain. Then for conductive deafness, it's caused by interference in conduction of sound through the outer and the middle ear. So it can affect hearing before the sound reaches to the cochlea. And then a foreign body or a wax is some actually one of the most common causes of conductive deafness. Then those are now disorders of hearing. What about disorders of now the vestibular apparatus? So these disorders for vestibular apparatus, one of them is vertigo, the other one is Menire disease, and then the other one is motion sickness. So for vertigo, is that this one affects the sense of bodily rotation. So this is due to the overstimulation of the crista ampullaris of the semicircular canals or the semicircular ducts. So remember, the crista ampullaris is the one that tells us the hair cells in the crista ampullaris and the macula are the ones that tells us the body state of equilibrium. So if you have now overstimulation of crista ampullaris, the hair cells in the crista ampullaris will have now vertigo, the sense of bodily rotation, increased body rotation. For Meniere disease, is that these are now episodes of vertigo accompanied with hearing loss and ringing of the ears, which is now the tinnitus, and it has various causes as we can see there. Increased pressure in the membranous labyrinth, and then this one can lead to rupture and leakage of the endoleaf into the perileaf, which is disastrous. For motion sickness, now this is overstimulation of the macula of the utricle caused by repetitive changes in linear acceleration or directional changes. So motion sickness usually get it. Some of us when actually we're traveling for long distances, that is motion sickness. And maybe now to the other um, special sensation is vision. And for vision, we know the eye is the organ for vision, and specifically the retina. So we can remember that the eye has three tunics. It has the outer tunic, the middle tunic, and the inner tunic. Outer tunic contains the cornea and the sclera. The middle tunic contains now the vascular part, the vascular cord, the choroid, the ciliary body, and the iris. And then the inner part contains now the retina. The inner tunic contains now the retina, the neuroretina, which is we'll discuss it when. So now the images perceived fall on the light sensitive membrane called the retina, the inner tunic. So the one in yellow at this point, that is the retina. So that is now the light sensitive membrane. It's light sensitive because it contains photoreceptors, which are now the cells that perceive um, vision. A point to appreciate is that we'll have some special parts of the eye, the fovea and the macular lutea, which now, at this point, the fovea is the area of greatest visual acuity, and then this point only contains cones, and then the region surrounding it will be called as macular lutea. Then the remaining structures of the eye, So talk about the vascular cord, the outer cord, the cornea, the sclera, uh, the aqueous humor, the vitreous humor, talk about the lens, the various ligaments, the various muscles, all of them. They either provide support in terms of to support the retina or to focus image of the visual world upon the retina. So that is a function of all, that is just to summarize the function of all the structures of the eye, either to support the retina or to help in focusing of the image onto the retina. The cell types of the retina. So there are three cell types, and the three cell types is that you have neurons, the pigmented epithelial cells, which is each increase visual acuity by absorption of scattered lights. Then also we have supporting cells. So remember, I had told us earlier, each and every special organ has supporting cells. So in an exam setting, if you don't remember any cell type, just write basal cells and supporting cells, and you'll be sure you're getting 2 marks. Then those are the three cell types, majorly the three major cell types in the retina. So neurons, pigmented cells, and supporting cells. For the neurons, their fathers are divided into 3 types. And the 3 types is that we have the major cell type that is supposed to appreciate today, the photoreceptor cells. And these photoreceptor cells are the rods and the cones. And then you have the cells of afferent fibers passing in the optic nerve. And then you have now integrating neurons. And these integrating neurons, they are found in between the photoreceptors and these cell types, the ganglion cells. The ganglion cells. And these integrating neurons are further divided into 3 types. That's the reason why I said you have basic 3 cell types, which are the neurons, pigmented epithelial cells, and supporting cells. For the neurons, we have photoreceptor cells, we have integrating neurons, and then you have the ganglion cells, which are now the cells of afferent fibers passing the optic nerve. And then we have now these integrating neurons being further subdivided into bipolar cells, horizontal cells, and amacrine cells. We'll see them in a few minutes. And maybe to summarize each up is that you can have a flow chart. So you have the retina, cell types in the retina. You draw 3 branches, neurons, pigmented epithelial cells, and supporting cells. Then, for the neurons, you draw below the neuron part, you draw 3 branches, one for the photoreceptor, that is the cones and rods. Then you draw integrating neurons, and then you draw now the ganglion cells. Under integrating neurons, you further subdivide into 3 bipolar cells, horizontal cells, and amacrine cells. So now for the supporting cells, we'll have molar cells and other cells that you'll see in a few minutes. So, and these are the layers of the retina. There are about 10 layers, 10 layers of the retina. So we'll start with layer number 10. So this is a layer number 10, as you can see here. The retinal pigment epithelium. So this is a pigmented epithelium, retinal pigment epithelium. Then you'll have supporting cells of molar. So these are supporting cells. Then the layer of cones and rods. And then you have now the outer limiting membrane. This is the outer limiting membrane. Then you have now the outer nuclear layer. So nuclear contains the cell bodies for cones and rods. Then you have a plexiform layer, which is a point of synapsing of now the cones and rods and the bipolar cells. Then inner nuclear layer now for the bipolar cells. And then you have the inner plexiform layer. And then this inner plexiform layer is a point of synapsing for the ganglion cells and the bipolar cells. And then we'll have now the ganglion cells, their cell bodies, giving us the inner. The layer of ganglion cells will give us now the fibers that go and form the optic nerve at this point. So this is the ganglion layer, the ganglion layer. So any layer which is known as nuclear contains cell bodies. Any layer that is called plexiform is an area for synapsing. So those are the 10 layers, but we'll discuss them further in a few minutes. So the outermost layer is the pigmented epithelial layer. So this is a pigmented epithelial layer, is actually the outermost layer. Then after that, now you'll have the processes of cones and rods, the photoreceptor layer. The photoreceptor layer, the layer of processes of cones and rods. Then you have now the outer nuclear. So the cell bodies of rods and cones will be found in this outer nuclear layer. Then the outer limiting membrane, I'm gonna press it here. Outer limiting membrane. So this outer limiting membrane now at this point will be separating the bodies of cones and rods and their processes. And then another point to appreciate is that we'll have the outer plexiform layer. So this is the outer plexiform layer. And then this outer plexiform layer at this point, you can appreciate this outer plexiform layer will be containing the dendrites and the axons of photoreceptors and integrating neurons, respectively. So at this point in this slide, we've covered 5 layers: the pigmented epithelium, photoreceptor layer, outer nuclear layer, outer limiting membrane, outer plexiform. But a point to appreciate is that the layer number 10 is retinal pigment epithelium. The layer number 9 is the layer of rods and cones. Then you'll have the outer limiting membrane, outer nuclear, outer plexiform. So we'll have this limiting membrane coming before the outer nuclear layer. For the other layers now, the inner nuclear layer will contain the cell bodies of integrating neurons. So these are the integrating neurons. Now the bipolar cells, And then the inner plexiform layer, so this is the inner plexiform layer, will be containing synapsing of various cells. And then these cells will be integrating neurons, the bipolar cells, and the dendrites of the ganglion cells. But a point of appreciation is that it will be containing some other cell types. For example, now the bipolar cells, which are these integrating neurons. These are the bipolar cells, which are perpendicular to the retina. We'll have horizontal cells in the outer plexiform layer. Then the inner plexiform layer will contain the amacrine cells. So bipolar cells will be contained, the cell bodies will be in the inner nuclear layer. Horizontal cells will be present in the outer plexiform layer. Amacrine cells will be found in the inner plexiform layer. Kindly take note of that and how they lie. So bipolar cells perpendicular. Horizontal cells, they are parallel, and then the amacrine cells are horizontal in the retina. For the ganglion cells, so this is a ganglion cell layer. Is actually a layer by itself, ganglion cell layer. So now the fibers from these ganglion cells will go directly to form layer of optic nerve, as you can see there. So the afferent fibers passing toward the optic disc to form the optic nerve at this point. The afferent fibers are coming from the ganglion cells, and then the last layer is the innermost layer, the internal limiting membrane or the inner limiting membrane. So by that, we'll have 10 layers of the retina. Actually, these are commonly asked questions. So be able to describe the various layers of the retina. But a trick is that being able to talk about the last layer is a pigmented epithelium. And then after that, we'll have a layer of cones and rods, then outer limiting membrane, outer nuclear, outer plexiform, inner nuclear, inner plexiform, ganglion cell layers, afferent fibers that form now the optic nerves, and then the last layer is the inner limiting membrane. You'll have formed all the 10 layers of the retina. So make sure you can be able to describe the 10 layers of the retina. So at this point, histologically, this is what you can be able to appreciate. This is the eye. This eye with the various tunics, but at this point, we just talk about majorly about this retina. So for the retina, higher magnification is that you'll have this. This is a higher magnification of the same. So be able to describe the pigmented epithelium. They talk about layer of concentrates. Then the inner limit, the outer limiting membrane. Then outer nuclear, outer plexiform, inner nuclear, inner plexiform. Then you'll have the ganglion cell layers. Then you'll have now the afferents, fibers that form the optic nerve, and then you finalize with the inner limiting membrane. So by that virtue, you'll have all the 10 layers. So pigmented epithelium, the layer of constant rods, outer limiting membrane, then you'll have outer nuclear, outer plexiform, inner nuclear, inner plexiform, ganglion cell layer. Then you'll have the afferent fibers that form the optic nerve, and then you finalize with the inner limiting membrane, as you can see there. So those are the 10 layers of the retina. Be able to describe this, take your time and draw it, and be able make sure that you can be able to interpret it at your own time. Maybe to finalize is that you're supposed to know various disorders of the light pathway. So starting from the field of view, the eye field of view, on how now you'll have the optic nerve, then the optic chiasma, then the optic tract, then you go to lateral geniculate nucleus, the optic radiations, and then you go to primary visual area. So be able to describe that pathway. In addition to that, be able to describe the various disorders. So for example, central scotoma, how does it happen? When you talk about monocular vision loss, that is anopia, how does it come across? Bitemporal hemianopia, which happens at the optic chiasma. You can also have now contralateral homonymous hemianopia. How does it come across? So at your own time, make sure you can be able to draw this pathway, the light pathway, be able to describe the various disorders that affect the light pathway, and then be able to describe what are some of the causes that can lead to those disorders. Yes, so that is another assignment. So remember, today I've given you about 3 assignments. You go and check the various differences between inner and outer hair cells of the spiral organ of Corti. We go and check and be able to draw the retina. Then now at this point, you can also go ahead and try to draw the light pathway, the visual pathway, and be able to go and draw the visual pathway and some of the disorders that can come across when you have now a lesion affecting either the eyes themselves, the retina in the eyes, or the optic nerves, or the optic chiasma, or the optic tract, or even the lateral geniculate nucleus, the optic radiations. So remember, for the optic radiations, you can have upper or lower optic radiation being uh, having a lesion that is a Mears loop and the rest. And then you can have now even the primary visual area being affected. So go ahead and check. And what do we mean by when we talk about contralateral homonymous hemianopia with macular sparing? How does it come across at our own time? So that is it. Maybe to finalize is that uh, there is this verse that always encourages me from the book of Colossians 4, that whatever you do, work at it with all your heart as working for the Lord, not for human masters. Since you know that whatever that you're doing, you will receive an inheritance from the Lord as a reward. And that it is the Lord Christ that you're serving, and it's not just mere human being. So during whatever you're doing, you just put our best foot forward so that you can be blessed. So that is it from me. Thank you for your time. And just to encourage us is that kindly continue sharing the videos and subscribing, and we're looking forward to sharing more and more content. So that is it. Looking forward to seeing you to the next video ahead. Thank you.